Thank you for joining our webinar about strategies for GMO testing. Today we have with us Pierce Smith from Eurofins GeneScan. Pierce has been with GeneScan since 2007 as a laboratory manager. His work focuses on the use of DNA and protein-based molecular technologies for the detection of biotech crops, as well as for authenticity, allergen management, and other food security related applications. We're very pleased to have him with us today to discuss this topic. But before we get started, we'd like to go ahead and review a few housekeeping items. This webinar is being recorded. We will distribute that to all attendees within the next 2 days for viewing. You can submit your questions throughout the presentation portion, and we'll address those at the end. Any questions that are not answered during the webinar, we can follow up on an individual basis afterwards. To submit your questions, you can go to the questions area on the dashboard here and go to webinar, type your question, and hit send. So with that, we want to go ahead and get started, and I will turn things over to Pierce, and we can begin the webinar. Sorry about that.
So does everyone see my screen now? Or do you at least see it, Heather?
Yes, see it here.
Perfect. So I'm going to be talking about PCR methods and analytical strategies for doing GMO testing. Basically, the topics I'll cover are: what are GMOs? Why do we test them? If we decide to test them, how do we detect them? And then sort of get into the nuts and bolts of assigning a percent or measuring a percentage GM. And how does that really work? So when we talk about genetically modified organisms, for quite a while, we've been tinkering with the different forms of life. We have viruses that can deliver drugs to cells. We have bacteria that are engineered to make things like insulin. Fungi we've sort of hijacked to make alcohol really efficiently. Plants, obviously, there's a lot of ag biotech, and increasingly there are some animals that have been tinkered with to grow faster or not produce so much waste and to be more efficient. Though by and large, when we talk about GMO testing, we're still really just talking about plants, so the ag biotech that's been on the market since the late 90s. So these genetically engineered crops that are commercially traded in the US and more or less around the world, corn, soy, cotton, and canola are the big four. Sugar beet, alfalfa, papaya, and squash, they're certainly out there in commercial setting, though the planting is not nearly as wide as the row crops. We have some discontinued products, so you may remember back in early 2000 the Flavor Saver tomato, which inspired a lot of love and dread in the media, and then there were some potatoes on the market for a while, but those were withdrawn, and you will see now there's an asterisk because any day now a new company is going to start marketing their own version of biotech potatoes that have low acrylamide and are resistant to some bruising. So in the very near future, potatoes will fall back onto the commercialized list rather than the discontinued list. Other crops that you may have heard about are rice and flax. These were inadvertent releases. So the trait developers were working on GM crops in these particular species, and for whatever reason, some of that seed got out into the supply chain unintentionally. In this slide, we're looking at both herbicide-tolerant and insect-resistant crops. Since the launch of the first product in the late 90s, you can see a pretty steep increase in adoption rates for GM, mostly because it makes farming a lot easier. And really the takeaway from this slide is that in crops where there is a viable GM option, overwhelmingly farmers are planting it. When you look outside of the US, you can see this is actually happening all around the world. Here in the US, we're the #1 user of biotech crops, but 2, 3, and 4, Brazil, Argentina, and India are all developing countries. So use of GM is not at all limited to the developed world. We tend to benefit from them first because most of them are developed here, but increasingly other nations are seeing the value. So this crop year, there are a couple of new events coming out of South America. In Southeast Asia, they're looking at some new products as well. So no longer is this just a US-Europe concern. It's truly global. So moving along, why are we testing for biotech crops? So in the US, a good rule of thumb is you generally don't have to test. And we don't have to test because the biotech crops and their products are considered substantially the same, substantially equivalent from their conventional counterparts. So this means corn is corn, whether it's biotech or not. And there's just no reason to test. There are a couple of exceptions, like every good rule. And the first one of those is for seed quality control. So the biotech companies that make these traits want to make sure that the seed they're selling actually has them. So they can do that in many different ways. If they have a herbicide tolerant, they just plant a bunch of seeds and spray it with their herbicide. And if it lives, guess what? The trait's in there. For things that are not quite so obvious, there are other options. You can use an ELISA test, which is enzyme-linked immunosorbent assay. That's a lab test that looks for protein. You can do PCR tests, which are DNA tests, and they can do sort of quick field tests, the lateral flow strips. And they do this to make sure that the biotech seeds are not in the bags that are intended for conventional planting, and also to make sure that the type of seed that's listed on the bag, if your seed is resistant to Roundup but not to a different herbicide, you don't want to get those things mixed up, because if the farmer goes and plants and sprays his field and everything dies, he's not going to be very happy that his very expensive seeds didn't resist the herbicide that they were supposed to. Exception number 2 is catering to consumer preference, either domestically or in export markets. So there are people who decide that they don't want GM food and they don't want to buy it. So to serve those markets, some testing is required. In the export market, you might be dealing with mandatory labels that say above X%. You must label if your product contains GM. And increasingly, there are voluntary non-GM labels that are not stipulated by law, but if a company is interested in producing food that meets the non-GM standard, they can just go through the process and do it themselves. For these sorts of testing, generally, depending on the product, a lot of lateral flow strips are used, also a lot of PCR, ELISA not so much. And the third exception is for asynchronous approval. So in countries that have laws about what biotech crops can be grown or put in food, there are regulatory systems where the trait provider submits a new crop saying this is what we want to sell, and the wheels of government turn slowly. So in country A, it might be entirely legal to sell a seed, but in country B, it's still illegal. And because there's no reason not to plant in the US when you're still awaiting approvals in the EU, this creates the situation where, unfortunately, some commingling might happen and you end up with an unapproved event in your shipment. So in these situations, even trace amounts can jeopardize certainly in the seed supply, trace amounts can jeopardize the whole crop, which is what we saw with the inadvertent release category for rice and flax. There are also discontinued events that were marketed for a while but then withdrawn, and markets are intolerant of those as well. And so in situations where even some small admixture of an unapproved event can block import, testing is critical to exclude these unapproved events. So what we're talking about here is not a safety issue, it's a legal issue, it's a regulatory issue, and a vexing one nonetheless. So when you come down to proving events, generally we're talking PCR test, which looks for the DNA rather than the protein. I'll get to that in a little bit. And it's important to mention that, food and food crops are tested pretty rigorously all along the supply chain. It's not just a GM situation. It's all sorts of measurements that are going on. So nothing particularly special other than what you're looking for. So now we know what we're talking about and why we test, getting into the nuts and bolts of that. This green cartoon is going to be my representation of native soybean DNA. And because I want to do something special with that soybean, I put together, through these little Lego blocks, a recombinant gene that I stick in there. And so this one is going to be the original Roundup Ready trait, more or less. And where that recombinant gene sits in the genome is what we call an event. Now, the biotech companies don't develop one plant and market that. They'll take that same insert and put it into hundreds or thousands of plants, and each one of those will have a unique event identification number that is specific to where it sits in the host genome. They'll take those thousands of plants and watch them grow or not grow, and the ones that perform well will be progressed in the commercialization pipeline. The ones that don't get thrown away and incinerated. And finally, they'll whittle it down to one, maybe two products that finally will go to market. But that's over the course of about 8 to 10 years. So once they have a marketable event, they grow it up in many of their elite seed lines. And in this cartoon, of a plant cell. Every cell nucleus will have a copy of this transgene, and we can use PCR tests to look for those gene copies that are in every plant cell. And we can do a qualitative test, which is just a positive or negative test. Is it there, is it not? Using similar PCR techniques, we can do quantitative testing, so how much is there? The genes that are in those nuclei code for proteins, and so as the plant grows and transcribes the genes into the protein, you can also use the ELISA test or the LFS test to detect those gene products. So two technologies looking at different lights, but to the same end. The difference between the testing really is that the PCR tests and the ELISA tests are lab tests, so they require specialized equipment, specialized training for staff, and generally a little bit more know-how. But the lateral flow strips, or strip tests, or lateral flow devices, those are rapid field tests. You don't really need any training. You need a grinder and a cup and some water. So these are used by the tens of thousands in every grain elevator everywhere that tries to segregate GM from non-GM. It's the first line defense, and the grain companies spend an awful lot of time and effort making sure that things they don't want going to silo A don't get there. And that's all done by LFS. So when we talk about the screening tests we do, these two cartoons represent two completely different crops. You'll see one is the soybean, the original Roundup Ready soybean. The second one is the corn product. And if you look at the little components, you'll see that we have a 35S here and a 35S there. So completely different crops doing different things have these shared components. And that's very convenient from a testing standpoint, because by targeting these shared components, you can catch a broad range of biotech crops without knowing specifically what you're looking for. And if you don't find these sort of permissive promoter and terminator markers, that's a very informative result. It catches a lot of different events with a limited number of tests. Of course, the trade-off there is that if you get a positive, because these are so non-exclusive, it's pretty difficult to tell what's causing it. But still a very good first line for crops and products where the expectation is there is no GM. Looking at this table, you can see pretty quickly how those shared markers actually make things a lot easier. In this column, you have a number of commercialized corn events, and most of those have 35S. There are a few that don't, but guess what? They also have this NOS terminator. So with those two tests, you catch all of the commercial corn currently. In soybean, you have 35S. You need FMV for some of the newer products. But rather than running dozens of specific tests, the markers provide good coverage with minimal expenditure for your testing dollar. Now, of course, there are situations where it's necessary to become more specific. As I was talking about the asynchronous approvals, if you have unapproved events, a yes/no on something that reacts to almost everything isn't that useful. So you look at different targets that are slightly more exclusive. So we can look at construct tests, which take our two cartoons that have the same 35S and the same NOS, but look at the junctions that they don't share, so between the 35S and the CTP4, which is not down here. You can discriminate between these two events. Or you can step back and go all the way to that event-specific test, which is the integration site into the host genome. And that will be the exact event. The problem here, you have to do one test for every event, and that gets sort of exhaustive and costly. The good thing about construct tests, occasionally there are entire classes of product that have a similar construct that will all be unapproved, and so by doing one construct specific test, you can knock out 4 or 5 different products that you don't want. Moving along to the quantitative part, generally speaking, you're looking at an event. So you're doing a single test for the biotech DNA, and then you add a second test for the plant genome. So this screen grab looks at sort of the raw data that you get out of a quantitative test. On the y-axis, we have signal, and the x-axis is more or less time. The way the reaction works, the more of a particular target you put in at the beginning, the faster you will see signal as it runs. So in these standard wells, we've loaded known quantities of our target genes, and as they run, they produce a standard curve. And using this standard curve, we can then plot our unknown samples along that line and derive values. So as I said earlier, this is really 2 different tests. So one is for the plant genome, the other is for the biotech gene. What you end up with, because what people are interested in is percent GM, is a ratio between the species DNA and the amount of biotech DNA. This is a relative quantification. That's an important thing to note, because we're not measuring how many copies of GM are in the tube. We're actually trying to compute a percentage of GM corn or a percentage of GM soy, not relative to the product as a whole, but just as a percentage of the corn or soy used. So common testing strategies, if you work in a regulatory environment or a research environment, it might make sense to do a single test for every single event out there. And you add all those single event tests up, you get a total percentage. Now, this is very accurate, but it's also very costly and very slow. And in a commercial situation, that's not useful. So, as I said earlier, there are shared markers, and we can do a quantitative test based on those shared markers as well, and lump together most of the products out there with 135S test. This is specifically corn we're looking at, but the same applies to soy and canola and the other guys too. And then add individual tests for the few events that the one marker does not collect. And then you get a reasonably accurate idea of what's in the whole product. It's a lot more cost effective, it's a lot faster. So some of the limiting factors in testing for GM, certainly in grains, are that sample sizes are what really determine the detection limits. So in grain and particulates, the amount of product you test is really what determines the sensitivity of the overall testing. It's generally not the technology that's limiting you, it's the sample itself. So on this graph, we have on this axis the true level of impurity, on this axis the probability of a negative result. And this is again based on a 3,000 seed sample. This curve would be different if you change that sample size. But you say, okay, I don't want to buy anything that's more than 0.1%. So here's your 0.1% impurity. And you look where that intersects your other axis. And this is how you derive the statistics behind this particular sample. So 3,000 seeds at less than 0.1% with 95% confidence. When you imagine that to get that 0.1% in a given crop, you have to test 3,000 seeds. You can see this is pretty variable depending on what you're talking about as far as mass weight of the actual product. So in corn, 3,000 seeds, about kilogram, but for something like flax or canola, an awful lot less. So if you arbitrarily set your sampling size at 1 kilogram, because that's the perfect size for a corn sample, you end up testing way too many seeds for these other small crops. So once you select the appropriate size sample for your crop, things get a little bit more complicated because, just like a canola seed and a corn kernel are not the same size, the actual genomes in those things have very little relationship to the seed size itself. And what you can see here is that rice has a tiny genome compared to wheat, which is absolutely massive. It's 2 orders of magnitude bigger. So what you end up with in a tube, if you sort of imagine rice is a BB and wheat is a basketball, if you have a mixed sample based on just the raw weight of DNA you're putting into your reaction, you don't get an even representation of what that product might be. And this goes back to why the relative quantification is so useful in GM testing. Because again, we're not worried about the content of what's in the tube, we're not worried about how much of any particular ingredient is in the whole product. Really what we're trying to determine is the percentage of GM compared to the host species that it comes from. So this slide talks about how processing a product can also affect the amount of DNA that's in it. If you start off with corn and you test for GM, you have a ton of corn DNA, a good amount of GM DNA, so that ratio of GM to donor DNA is very easy to do. You have 1% with a very low LOQ, so based on this 3,000 kernel sample, which we know informs that LOQ for this particular sample, we have a very specific number. Now, if you take corn flour, you get a similar recovery of DNA and a similar number. So 10,000 copies of corn to 1,000 copies of GM DNA, still 1%. This LOQ, however, is much lower. That's because up here we're testing 3,000 kernels, In corn flour, in a commercial lot, you have hundreds of thousands of kernels. So this is really more the technological limit of detection, whereas this one is a sampling limit of detection. So both corn and corn flour are relatively unprocessed. They have lots of DNA and lots of target. If you start turning them into other things, however, corn gluten, these numbers are even just for illustrative purposes, but tenfold less DNA in corn gluten than you have in the corn that it's made from. But that's still a good. You have good statistical representation, so you can apply a nice 1% result at a low LOQ. However, when you go from corn to something like corn starch, the production process that makes the starch that you want to sell and use also washes away almost all the DNA. And this 1,000 copies of corn in the starch sample is probably a native starch, so completely unmodified, pretty crude, and you get a result that, yeah, somewhere around 1%, maybe we can say, but also this 1% is sort of the cutoff for commercial viability, so not really a quantifiable matrix. Into something highly refined like maltodextrin. You start with 100% corn, you end up with almost no detectable DNA at all. Maybe you get a positive hit for GM, maybe you don't. Again, LOQ increases, still not a quantifiable matrix. Something like corn syrup, the sugar-making process is very destructive and it has a lot of cleaning steps. So you can start with 100% GM corn, turn it into corn syrup, and there's absolutely nothing left to measure in the end. So no PCR test anywhere on the planet is going to say, did that corn syrup come from GM corn or not? And this gets into why final product testing is often not as illuminating as people would like it to be, just because the processing has completely removed all that target analyte. So to qualify a corn syrup product as non-GM, you can test it 6 ways from Sunday, it's always going to be negative. So there's no smoking gun, but really the key piece of information there is to be able to link the syrup that you're putting in your bottle back to the actual corn that you ground up and made it out of. So this pie chart describes a product that I certainly would not want to eat with corn and wheat and pea flour and rice. It doesn't sound very tasty, but it does illustrate a vexing problem that industry faces. So in this product as a whole, you only have 1% corn. That 1% corn is 100% biotech. You just bought it off the open market, did nothing to control for it. So, if you ask a guy on the street or an analytical chemist how much GM is in this product, it's not an unreasonable answer to say 1%, because of the product as a whole, it's only 1% of this GM corn, and by and large there is not GM rice or peas or wheat. However, when you do the DNA test, remember we're talking about a relative quantitation, so there's no way to calibrate to the whole product. And when we look at the amount of GM in corn relative to the amount of GM in the product, that's going to come back at 100%. So that's just because the ratio of corn DNA in the product to the biotech DNA is stable. But it says nothing about its relationship to the product as a whole. Why is this useful and interesting? Well, say you take the exact same corn and you put it into a different product that instead of having wheat flour with a lot of DNA has sugar, which has very little. If you do a quantitation of corn DNA or biotech DNA to corn DNA, you get the same number in both of these products. However, if you try to work out a system where you calibrate to the amount of DNA that you see in the product as a whole, because this matrix has considerably less DNA, the same amount. So same product in a different formulation gives you a whole different number, and that's not useful for anybody. Here's another interesting case study that sort of illustrates the same point. A customer makes a feed product and they ship it, get a lab result back that says, oh, hey, this feed product you have has way more than 10% biotech corn. And then lab A, as it happens, is the regulatory lab in the EU, so they say, you can't sell this or you have to put the sticker on it. And the company says, okay, well, let's see what a different lab says, and so they send it to a different lab, and turns out this lab comes back with, yeah, way more than 10% GM relative to corn. If you just look straight at 35S, it's somewhere over 100%. So in this relative amount, all the corn is GM. So, but wait, we have this other lab that's saying they're testing 35S, which is exactly what lab B tested. And it's saying less than 0.1. So what we're really looking at here is a question of calibration. So the regulatory lab is using a relative quant. This service lab is also using relative quantification. But this other lab over here, they're just doing an absolute measurement of the amount of 35S in the tube. And because that seems quite low, it leads to a false sense of security. Again, when you're shipping to a regulated market, it doesn't matter that you have this nice result that says, oh, we're in spec. All that really matters is, do your tests match what the regulator is going to use? And I would say, yeah, if this is what the regulator is using, everybody's happy. But in the EU, they definitely are not. So another reason why absolute quantifications are not particularly commercially useful. So in summary, the most commonly used test for GM and food products is PCR. And currently, you get very good coverage using the combination of the 35S promoter, the NOS terminator, and the FMB promoter, because they're widely used in corn, soy, canola, and a number of other crops. Over time, that will be changing, but as of today, we're still pretty good with this setup. If you're sending your product to an area where there's risk of unapproved, more specific testing is generally advisable. And when you try to apply results for PCR tests, or any test from that matter, to a complex or processed food, often the number you get isn't as meaningful as it might seem, and there's a lot more paperwork that needs to go behind just putting a non-GMO sticker or a non-GMO label on the product. So I think that gets me through my canned talk, and I will turn things back over to Heather to tell me about any questions that might have popped up while I was blathering on. And
Great, thank you, Pierce. We do have a few questions that came in. First question, you just were talking a little bit about some differences possibly in the testing between U.S. versus EU, and they're wondering if you can talk a little bit more about what those differences are and what is driving that difference.
So if we go back to this slide, this literally is a real life example, and Lab A was the regulatory lab, Lab B was my lab, and I don't remember who Lab C was. The differences driving these tests are literally the laws in those countries. So when the EU back in 2003, 2005 finally worked out their legislation, they said, this is how you define GM. And so they only accept relative quantifications because that's what their law says. Now, when you talk about Japan, they also came up with a relative quantification system that they stipulate by law must be used for corn. They, however, set up their test a little bit differently. So the genetics aside, the way Japan measures GM corn is not the same way that the EU measures it. Even though they're both relative to the corn donor, you'll take the same batch of corn, apply an EU test and a Japanese test, and get about a twofold difference. So this really is a problem for industry. Everybody recognizes that, but since these definitions are enshrined in law, and at this point pushing 20 years, it's really difficult, even when everybody's in the same room and more or less on the same page about wanting to be transparent and to not put up artificial trade barriers. Getting the people in the room to agree that there's a better way forward is well and good, but getting the governments involved to recognize those recommendations and change their existing laws is just not reasonable. At least currently. Maybe down the road it'll all be such a headache that we'll all throw up our hands and say it's not worth it, or this is how we're all going to do it. But it's really a matter not of technology, but of the way the regulations are written.
Okay. Next question is, what new traits have been commercialized for planting for the 2015 crop in North America?
So the big new one in corn is Duracade, the Event 5307. It's definitely in the ground and does not have 35S, so that's the biggest new player on the block. There's also a drought-tolerant event. The number 87460 is also being grown. It does have a 35S, so not a big change there. It's also approved in the EU, whereas Duracade is not, I think. So not a big change this crop year. Every time we look at plantings, we think, oh, next year's going to be the big one when there are 5 new soybean events. But so far, so good for the soy guys.
Okay. Next question. Does quant digital PCR provide lower sensitivity for partially degraded processed foods than regular RT-PCR?
So the digital PCR is getting back into that more absolute. How many copies can we detect in the tube? So to that point, when you compartmentalize, is not really commercially valuable because, again, the regulatory labs aren't using digital PCR. They don't have digital PCR standards released, and you can argue till you're blue in the face that this number is valid and great, but tell a regulator he's wrong, and you're not really doing yourself any favors. So maybe somewhere down the line, again, digital PCR will offer a valid alternative to the relative quant, but currently it's just a different number. Honestly, it's no better, no worse, but not recognized in regulatory trade.
All right, I have just one more question that came in. What is the current regulation requirement for GMO labeling? I think the question was in relation to how it's presented, non-GMO versus GMO-free.
So GMO-free, at least in the U.S., is illegal. If you call something free, there's a reasonable expectation to expect zero, and you can't test for zero. It's just technically not possible if you want to have any product left over for sale. And when you step back from GM-free and get into the non-GM, things get murky again, because just like the government set their own standards for testing, they've also come up with their own levels of what is and what is not acceptable. For several hundred years in the grain trade, 95% was considered a good number for a bunch of quality standards. So based on what the industry was using for protein content and those sorts of things, Japan said, well, we'll say anything less than 5% counts as non-GM, because this fits neatly into the sampling statistics that we already use for grain. The EU, on the other hand, said, well, 5%, that sounds like an awful lot to us, so we're just going to say less than 1%. And then somebody in the room was like, well, less than 1% would really be better than just 1. So EU is by default 0.9 because it is less than 0.1. If you go to Australia, they said, well, we're okay with that 1%. So 1 is the line in Australia. Korea, I think, is 3%. The US, where we don't have a regulation, you're sort of up in the air. Companies can either define that number themselves based on what works for their own product, or if you're lucky enough to already be exporting to a regulated country, just take your regular product line and say, I'm shipping to Japan, I don't have to label it less than 5. That's what I'm going to use. And you're more or less good to go in the US, unless you are trying to apply for a third-party non-governmental standard, which may say something different. And currently there are 2 or 3 operating in the US. I think the biggest one by far is the Non-GMO project, but there are a few others out there that may be subtly different. I guess the takeaway there is your mileage may vary, but a low percentage, somewhere between less than 1% and no more than 5%, gets you where you need to be in almost all situations.
Okay. That's all the questions that we had. At this time, I'd like to remind everyone that the webinar has been recorded, and we will be sending that out so that you have access to the recording to view again. If you have any additional questions that come up, please feel free to contact us through the contact information that was provided in that closing slide. Otherwise, thank you very much for joining us. We look forward.
Go back to that closing slide. So
there you go. There's the information. So again, if you have any questions, please reach out to us. And thank you very much.